Microbe TV. This is TWiEVO. This Week in Evolution, episode 113, recorded on the 23rd of April in the year 2025. I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent, great to be with you. Good things come to those who wait. We're a minute or two behind on the live stream, but we're up and running, cranking up the engines yet again. Lucky episode 113.
That's right. Moving
Yeah,
along.
moving along. Good to be together. It feels like, once a month feels like once a year in terms of all of the action that sort of is unfolding scientifically and otherwise these days in our country. But good to be back together. And today we are going to pick up a piece of science that has been in the news headlines, ripped from the news headlines, and try to do a little kind of behind the scenes of what's going on here. Probably a lot of folks who've are listeners of TWiEVO are also have read some of this and are weighing what some of this means.
Before we get started, let me thank our moderators for being here today. Tom is from Western Oregon. Here he is.
Hi, Tom. Welcome back. Good to have you here.
Les is in California.
Hey, Les.
Barb Mack is in the UK.
Great. Good to have you back, Barb. Thank you.
And that is our moderation crew today.
Team of experts.
Welcome to everybody else. Tell us where you're from.
Yeah.
We will shout you out. Abdul Aziz is from the Kingdom of Saudi Arabia, where it's 30 C. That's pretty warm.
Ooh, quite warm. Welcome back.
What do you have over there, Nels?
We're kind of easing into spring. So about 60 on the Fahrenheit scale. And cloudy today. It's been sunny and kind of swinging between winter and spring. Can't decide which way to break, but I think things are warming up. How about there on the East Coast?
22 C here in Chelsea. It's sunny now, but it's going to cloud up later when I go off to teach my class.
Oh, nice.
Echo. So Les says
we're
there's
echoing.
an echo.
Uh-oh. Let's take a minute to go under the hood here. See if we can uncross some wires.
Why is it echoing?
Good question.
Okie dokie. There shouldn't be any echo.
Uh-oh.
Because I'm not sending back.
Audio?
Maybe I am. Let's try turning off that. Can you still hear me, Nels?
I can hear you.
Now I don't hear you. So I need to have
You
that
lost
on.
me. I could hear you that whole time.
Hell, where is the echo coming from? Didn't change anything since last time.
Gremlins are in the machine, as they say.
Oh, okay. So Les said he had 2 streams running. I'm okay.
Oh, well, it's on Les's end. Okay. Whew.
So thank you for letting me know.
Yeah, good to get the feedback. Good to get the feedback. Even better when things are okay. Yeah.
Let's see who else. Jan is in Tucson, Arizona.
You're welcome.
Ankley is in Norfolk, England.
Great.
John is in Eden Prairie, Minnesota.
Good to have you back, John.
Gary Weiner is in Denver, 50F.
Welcome.
Ms. Martian is in New Mexico.
Great. Thanks for joining us.
Jose Misleredo is from the Salt Lake City of Nakuru at the Great Rift Valley in Kenya. Wow.
Awesome. Fantastic.
That's very cool.
Yeah, welcome.
Alexander is in Paris. Very cool, Alexander. Good for you.
Great. Yeah, great to have you with us.
Rob said, dogs are evolving at pace.
Have we tipped our hand of what the topic of today's TWiEVO
Yeah, you
is.
did.
We're back.
You
Oh
did.
yeah, we're back in the canine category here. The dire
Steph.
wolf. Hmm.
Steph is in San Francisco. Our moderator. Thank you so much
Yeah, great to have you
for
here.
joining us. My audio is low. You can hear me right now.
I can hear you loud and clear.
Let's
Um,
see. What's the start? My channel 3,
mhm.
and I could crank it up a little. Channel 3. All right. Hello, hello, hello.
I can definitely hear you there.
I turned it up 2 notches on the channel strip.
Okay. We'll see if that does the trick.
Heil 1. The name of this microphone is a Heil, Heil, which is unfortunate, right?
Yeah, so allegiance to the microphone. I've got an Audio Technica over here. This is the microphone you recommended to me
Yeah.
way back, sort of in our opening um, attempts at TWiEVO, early days, probably coming up on 8 years ago now.
Well, yeah, I was just finding some old files. I found TWiEVO 5-year thing where we got people to record their stories. Remember?
Yeah, that was really fun. Got some
Mm.
evolutionary folks to give us a shout out. That was great. Yeah.
Yeah, I had going through hard drives and deciding what to keep. I can't keep everything. I don't have space. I have like
Yeah, I hear you.
50 hard drives here, and I don't think I have to save everything.
No,
Right? I just
we even
save
were. Yeah.
the raw files and then the final video, and that's enough. I don't want to save like the
All the channels and so forth. That's
no.
enough. We used to be sponsored, Vincent, by a hard drive company. I forget what they were called, but they even.
Drobo.
That's right.
Oh yes,
Drove.
that was Mark Fuccio who used to work for them,
Uh-huh.
and he shows up
Yeah.
now and then here and on the live stream.
Cool.
And he had he'd been listening to TWiV, and he said, "Why don't we sponsor you?"
Yeah.
So he gave us a couple of drives,
Hmm.
and we had a contest. We gave some away, and one year at ASV, someone came up to me and she said, "Vincent, I got one of those drives from you. I'm still using it."
Yeah, you sent one our way too, and we had it chocked full of vaccinia virus genomes, long read sequencing, early days
Cool.
of Oxford Nano. So yeah, it worked well. We were also sponsored back in the day by Blue Apron.
Hmm.
Got some good meals out of that. Is Microbe.tv still in the sort of sponsor
No,
attracting business, or we've let go of that
no,
as
I
a,
stopped years ago.
yeah.
I
Yep.
decided. So we had hooked up with an ad agency in San Francisco,
Mm-hmm.
and they sponsored us. We made some good money from that, and then we had like Blue Apron. We were one of the first people to have Blue Apron, and I was amazed at all the scientists who loved it. You know,
Yeah, absolutely.
we had Curiosity Stream,
That's
a
right.
streaming service of mainly sciencey stuff.
Mm-hmm.
But then the ad agency just said, "We can't make enough money off you. We're throwing you out the door." And so I said, I don't want to do ads. Screw this.
Yeah.
It's a real problem. So if you have an agency, they do all the
Mm-hmm.
work for you,
Mm-hmm.
but they take 30%
Yeah.
of your revenue, which is a lot, but they
It's
do
a
a lot of
lot.
work because they have to find the advertisers, they have to pitch, they have to then give a script, and then they have to invoice and get the money. And I don't want to do that. But more to the point, I decided for educational programs, I didn't want advertisement interrupting
Yeah.
them.
Yep.
And I don't even have ads on YouTube.
It's
Although
great.
I did a calculation recently.
Hmm.
I wouldn't make a hill of beans by turning on ads. It would
Ads.
just be annoying. I mean,
Agreed.
even though we have lots of subscribers,
Yeah.
you need to have a million to make a difference. So
Yeah. Yeah. Yep.
that's the story.
Different.
So no more ads. We are depending on our listeners. And you can go to
Exactly.
microbe.tv/contribute if you'd like to help us out.
Yep.
Um,
Absolutely. Yeah, I'm with you. Good call.
we
The
have another moderator here, Vanity Nutrition.
Oh,
Welcome.
great. Fantastic.
Good to
Yeah.
see you.
All right, we're firing up the engine. So let's dive in. Vincent. So, ripped from the news headlines, this is the dire wolf, of course, this splashy story about de-extinction. We'll kind of try to go beyond the headlines into what this really means, and to really, unpack the science here and try to weigh that contribution relative to some of the, the attention, the spin, sort of the, speaking of kind of ad agencies or kind of promotion PR
Mm.
operations here. That's definitely a big part of this story. But the reason I think we could, really get our kind of mitts into this or get our claws into this is because, so this is I'm guessing most of our listeners are already familiar, given the sort of widespread press here. But the reason we can kind of dig our claws in is that they actually did finally post some related work, a preprint on the bioRxiv, and so we can, look at that data, weigh that, and see where we go. A little bit of this might be a little too much provocative, but a little bit of a bait and switch. So the preprint that we'll be talking about talks about their new work on getting sort of paleogenomics. So getting bone samples of extinct species, in this case, the dire wolf of fame from what is it? Game of Thrones. I think it's. I don't watch Game of Thrones. I have to sort of disclaimer there, but is a prominent character, so sort of a familiar beast that has come into popular culture. They're going to do some genomics on this, put it into its evolutionary context, and then actually ask or try to get a sense of what are some of the genetic changes that might be adaptive in the history of the dire wolf, which then certainly sort of fed into the pipeline that they used. And this was the quote unquote de-extinction of the dire wolf. So they've have a few genetically modified clones of gray wolves that are now running around in an undisclosed location in the US, 3 pups that are growing up. But this kind of goes to the water's edge of just doing the genomic analysis, but doesn't bring us all the way to the actual advances they did in cloning. And
They don't
so,
even they don't even mention it, right? In the paper, in this
no,
preprint, they don't mention it.
correct. And so that's a little bit, I was kind of following it just at a distance here. And so, and I think this actually illustrates the whole, we're busy people, right? All of us are engaged and we sort of tap into our news sources, but it's really hard to go deeper and spend the time here. This is sort of, I think, the advantage of TWiEVO, other podcasts, of Microbe TV, is that we take the time to kind of go beneath the headlines and see what happens there. But you're exactly right, Vincent. As they said, oh yeah, no, we'll make this available. And there was a little hiccup with the press releases that were released before the preprint. And they did release the preprint, but the preprint is only, kind of gets you to the starting line of the really sort of provocative science here, which is the new wolf pups that are running around, and what does that mean from sort of a genetics
Here's the
standpoint,
here's
technology standpoint?
here's the authors and the name.
Yeah, exactly. So the preprint here, this is from Colossal Bioscience. So on the ancestry and evolution of the extinct dire wolf, but notably in the title does not go and then the quote unquote de-extinction. But let's step back and sort of get a look at the players here. So there's 2 co-first authors on the paleogenomics, the sequencing of the DNA that was recovered from some bone samples of dire wolves, and we'll talk about that in a few minutes. But Gregory Gedman, co-first author with Kathleen Morrow Pirovic, and they're both employed by Colossal Biosciences, headquartered in Dallas, Texas. We'll talk a bunch more about Colossal as well. Sort of long list of authors here. I won't go through everyone, but and as usual, an international collaboration here, people working at sort of all points on the map and around the world. So from Texas to Santa Cruz to the Smithsonian in DC, all the way to Copenhagen, Denmark, Oxford, UK, Princeton, Trondheim, Norway, University Museum there, Harvard, MIT, sort of a long laundry list. Again, underlining how modern science is practiced in international ways, collaborative ways, even in cases here where it's put forward by a biotech company, Colossal Biosciences. And so the chief science officer of Colossal is Beth Shapiro, the senior author on the paper. She, up until recently, was set up in Santa Cruz and helped to launch a sort of paleogenomics initiative there, and then it's grown into this more recently. This sort of spun off into Colossal. Also includes, so Ben Lamb was was the founders of Colossal. George Church, sort of a well-known geneticist from Harvard, who's done a lot in this space, cloning, bringing genetics to sort of biotech sort of angles. And then here's a notable author on the list. Vincent, did you catch this? It's George R.R. Martin.
No, I didn't catch that.
Yeah, so fourth to last author. That's the author of this paper, also the author of Game of Thrones. And so tying together
Wait,
sort
the
of
author of the books.
correct. That's
Oh,
right.
I wouldn't have known that. I
Okay.
wouldn't have known.
Yeah.
I've never watched a minute of it.
I haven't either, but we're sort of exposing ourselves as sort of out of the mainstream popular culture here. But anyway,
we had an episode of TWiEVO called Game of Clones.
that'd be a good title for this one. I think I've got our current working title is A Wolf in Wolf's Clothing. We'll see where we end up. And
we had a person on, Ben. He was at NYU at the time. You know him. But anyway, he had done a cloning game with people, and so he came and explained it, and so we called it Game of Clones. It was good.
That's great. That could be a working title for this one too. I think we'll circle back to that idea. So, Colossal Biosciences was founded or opened in 2021. It's a relatively new company, about 4 years old. I think it has locations in Austin, Texas, Dallas, and Boston. And currently, I think it's at roughly 130 scientists working there. So relatively kind of medium-sized outfit, growing. American biotech and genetic engineering company working to de-extinct several extinct animals. So this includes woolly mammoth, Tasmanian tiger, northern white rhinoceros, which has also been in the news headlines recently. I think down to just a small handful of individuals in that population, just on the edge, the brink of extinction. The dire wolf, which obviously we'll be talking about today, and then the dodo bird is also in the catalog here. So, needless to say, and probably a lot of people are already familiar with at least some of the controversy, this idea or this notion, the definition of de-extinction. Are we really, in these cases, bringing back a species that's gone extinct?
Well, now, let's say we could bring back an extinct species, right? That obviously you'd need the entire genome sequence, and then you'd have to do something with that. It's hard, right?
Yeah.
What if we could do it? Should we, or should we let evolution stay where it was?
Yeah. Well, so, and first of all, just having, so this is, you've already, I think, touched on one of the disconnects here, which is having a whole genome and then putting or breathing life into that genome. So actually bringing back that species is not what not their actual goal here. So when they say de-extinction, they don't mean to bring back sort of that entire kind of snapshot of the genome at that point in time when a species went extinct, but instead to bring a few sort of key genetic changes that then on the sort of framework of an existing species, in this case will be the gray wolf, and we'll unpack this, but it's only 20 genetic changes and among what are inferred to be millions of genetic changes. And so this is a genetically modified gray wolf that we're looking at. Now, of course, they've picked some traits pretty carefully to try to, hit on some of the sort of charismatic features, the size, appearance, the, thick white coats versus the gray wolf, sort of more modest jacket, and things like that. And again, that sort of PR, that sort of provocative nature of it. There is underneath this, though, I should say, it's not just sort of the, kind of visual or morphological traits. The notion here is also to try to fill, niches that are unbalanced because species have gone extinct. So, when we think about ecosystems, if you take away some of the key predators or at different sort of links in the food chain, that can really have a huge impact, knock-on effects basically on all the other species there. But this unpacks a whole nother problem, which is, okay, well, if that habitat has been lost,
Mm.
or the sort of parts of the food, if you bring something back that sort of fills some of those spots, is it too late? Is there, or, or what are the prospects for sort of rebuilding the fabric of biodiversity that's sort of necessary in these cases? And so,
Well,
um,
so also it could be an invasive species, right?
absolutely. So that also really comes into play here. So I was doing a little bit of digging, and we'll continue to touch on this during the episode on the website for Colossal. I think, to me, actually, the most beneficial thing they've done so far is they developed a few vaccines for viruses of animals that you find in zoos. So
Mm.
there is that's maybe the most practical outcome of the sort of the resources that they've had so far. But then, of course, the real sort of central mission is this notion of cloning animals, of bringing back genetically modified animals that sort of approximate what some of these extinct species looked at, develop that technology in the hopes that it might be deployed in ways that are
Mm.
ultimately positive. And so I think that might be sort of, part of, or one way of framing this, the whole thing is it worth it? Kind of all of that energy that they bring to the table here, and the resources, the eyeballs that they command as they sort of, are doing these glossy coordinated pieces in Time Magazine, et cetera. Does bringing those resources or that attention to this work then lead to scientific advances that then might be deployed in different ways? And so, in that sense, if we really step back, this idea that we kind of develop technology as humans, as scientists, and then try to use that to solve some of the current problems, create new problems along the way, and then sort of try to chase that with new technology that then solves the problem, and that sort of kind of leapfrogging from stone to stone that we see in a lot of fields, whether it's biological sciences, engineering, etc. So anyway, and speaking of that, in addition to the kind of the resources generated here, there's also politics, I would say, involved in this. So Colossal also coordinated to some degree, or got on the radar at the Department of Interior. So this is now run by Doug Burgum. He's a former governor of North Dakota, now the Interior Secretary. And so with the report of the 3 dire wolves, the Department of Interior went on the record to say how excited they are about this technology, how it might serve broader purposes beyond recovery of lost species, which again is, we'll unpack that some more, strengthening of biodiversity protection efforts, and helping endangered or at-risk species. So again, a sort of a bit of a bait and switch here. And in fact, if depending on your reading of this, some of the interpretations of what's coming out of the government now, and what's happening in terms of the Endangered Species Act, it's really being weakened, in part saying, oh, well, we have all this technology now, and so we don't really need to protect endangered species. We'll just, if we lose some along the way, as we're sort of letting go of some of these protections or regulations, there's, on kind of the extreme end, is this idea that, oh, we'll just develop some new technology and de-extinct a bunch of stuff and we'll be fine. That's pretty glaring, and I think for me, a pretty scary prospect, given the disconnect between what it means to recover lost species and what's going on here, including, I think at the EPA, there's news now that they're trying to say that so imagine an endangered species, right, that has a limited habitat. A lot of the habitat has been lost to human activity. They now want to reclassify the loss of habitat as not a harm to species, not a direct harm to species. And so the real danger here, I think, is as we see the technology advance, is that then the government sort of steps away, or sort of puts this in its place, and you're not patching a hole here. You're probably making the problem even bigger. At the same time, it's not kind of black and white here. It's these nuanced shades of gray. And so is innovation a good idea? Is bringing some genetic technology to thinking about biodiversity, right? Like, this is what we've done all along, is as humans, we've intervened and tried to, as we disrupt things, we also try to, on the other hand, use technology and human advances to try to make things better. So it's sort of, again, this leapfrogging game of developing technology, new problems, and then trying to solve them as we go. Okay, so that's kind of maybe a little bit of the backstory here, the news headlines. But then now let's kind of dig into the actual genetics and see where we land, or where we end up. It'll be great to get. I'd love to get, if people want to sort of chime in with questions, comments, reactions, different angles on this as we were all sort of processing some of this information. That would be really fun to bring that into the conversation as well. So what the scientists did here, as opposed to bring back the dire wolf, is they made 20 genetic changes to 14 genes in the gray wolf genome. They used gray wolf blood cells, placed them into those, sort of modified those cells, and then launched them using donor eggs that were carried to term by dogs, so not actually by wolves. So the mothers of these genetically modified wolves are large dogs. So again, so this part of the work, sort of the most provocative part, the genetic engineering of these clones, genetically modified clones, is not on the record. So it's hard as scientists to really dig into exactly what they did. And so now we're depending on sort of science by press release or science by news story. I haven't done a massive deep dive into this, but the Time magazine piece, which is a coordinated effort, the writers at Time were given access, reporters at Time were given access by Colossal to the wolves and some of the backstory, the parts that they wanted to share with the reporters. And so in that article, that sort of splashy, extinct with the cover of Time with extinct crossed out, and the picture of the gray wolf, they collected blood cells. So these are endothelial progenitor cells from the blood. And that might mark the biggest advance here in terms of the genetic engineering. So up until now, so Dolly, the sort of famous sheep that was cloned 30 years ago, they needed, instead of using blood cells, it was more invasive. They had to use some of the tissue and different tissue types as they launched a genetic clone of Dolly the sheep. Here, you can just take a blood sample, in this case from gray wolves, and given these the subset of cells, these progenitor cells, they're able to use those to sort of launch their new genetically modified animal. And so these are cells which would otherwise form the lining of the blood vessels, but they're reprogrammed to become germ cells that then can contribute to the birth of a new animal. So then they, in addition, so they collect those cells and then they use some genetic engineering. Again, we don't know the actual details of this, probably CRISPR-based gene editing, but they make changes to 14 genes in the gray wolf genome. And then and they use the preprint here. They use the information that they got from the dire wolf genome to make some predictions about what are the genes that might have the traits that they're interested in exploring here. So no ancient dire wolf DNA was actually spliced into the gray wolf genome. This isn't Jurassic Park, but the modification or the mutations that they were able to sequence and annotate, those were used. And then the sort of genetic editing technology. It's basically a guide RNA that has some changes. You bring this CRISPR Cas9 system in the protein, finds that region of the genome, that gene, and then that template, it changes it, edits it a little bit. And so there's some sort of different flavors of CRISPR engineering. I'm assuming that's what they're doing, but again, these are sort of trade secrets in a sense. So they haven't given that away.
So they're doing this on gray wolf DNA.
Exactly. Yep. And making those 20 changes to 14 genes. And then they still have those cells. So these progenitor cells that they've reprogrammed. And then at that stage, they take the edited nucleus and they transfer it into a denucleated ovum, an egg cell, donor egg. Right. And so this is nuclear transfer. The genetically modified cells, the nuclei are taken out, put back into, and then that egg then develops into actual embryo. And it's brought to term by, in this case, a dog mother. And again, we don't have all of the details there as well.
So along with the desired changes, there are probably some off-target changes that they don't know about, right?
Absolutely. Off-target changes, we don't know about that. And you'd have to, maybe they have done that so that you could do an entire genome and sort of try to ask, what are those? That's probably underlies so the efficiency here, again, this is kind of back of the envelope calculations, maybe it's 1%. That might be generous. Effectiveness, that could have been some of that low number. So it's a big technical feat to pull this off, to transfer a nucleus into into an egg, get it all to develop and go. So I'm not trying to downplay sort of the degree of difficulty or the technical advance here. But why is it 1%? In part, it might be some of those edited nuclei. There's all kinds of, just like you're saying, Vincent, off-target effects that would then not lead to a viable embryo because it's sort of this mutational weight on there. So I think for each of the three, again, the numbers are hard here, but they did so 45 engineered ova per ultimate wolf or something in that neighborhood. They're developed in embryos in the lab, and then it's the embryos that are inserted into the wombs of two surrogate hound mixes, chosen mostly for health and size. Can they sort of handle a wolf as it's developing? Um, and I think that these are like C-sections, so they're not bringing the wolves to term. And then they said, so the births were in October 1st, 2024. They got 3. So this is Romulus and Remus. I think that is referring back to Game of Thrones. Maybe that was like George Martin as an author on the genomes. Maybe that was his contribution, was the
Founders
the
of
names.
Rome.
That's right. Exactly.
Fed by a wolf. They were fed by a wolf.
Exactly. Oh yeah, absolutely. This is all tying back to the mythology. And then a few months later, Colossal repeated the procedure with another clutch of embryos. And then, so this was January 30th. I think the first 2 dogs are males. And then the third one, Khaleesi, is a female. And obviously they want, as they're thinking about kind of taking this forward, we've got sort of issues potentially with obvious issues with inbreeding, but then to start to get a pack going here, right, would be sort of one of the ideas. And so
why do they want to do that?
Well, so I don't know if they, so the bigger, all of the kind of disconnects here, right, or the bigger picture, I think on the one hand, they want to advance cloning technology in animals.
Mm-hmm.
That's sort of, in actually, and that, again, that's probably the advance here, is being able to do this from blood cells. So now imagine doing this with cattle or sheep like Dolly, et cetera.
Sure. Okay.
Lowering those barriers. There's sort of less sort of provocative impacts here on things like agriculture or livestock genetics potentially.
Yeah. Okay.
So that's that's part of it. But then I think if you sort of listen to Beth Shapiro, who's chief science officer and who has been so they've, maybe one of their bigger advances up till now was the woolly mouse. So they have the woolly mammoth on the menu. Again, taking modern elephants, doing sort of a similar procedure, and sort of as a proof of principle, they did the woolly mouse. And so very similar, they pick a few genes to make mutations and then get that sort of, charismatic phenotype or charismatic trait. Here it's that woolly fur, thick fur on a mouse. And so they're hoping to do that with elephants as well. And then if in weather, then you create an animal that can then, go back to Siberia for, if it's a woolly mammoth, or the dire wolves in sort of northern North America, and start to fill these niches. People have pointed out, and we'll get into some criticism as well, that, if you really did sort of continue to advance this pack of quote unquote dire wolves or modified gray wolves, what are they going to eat? And so you're kind of setting up, as humans intervene as well, how I think one of your original points, right, is like we sort of let evolution moves on and things adapt more naturally. How much sort of input are humans going to put into this to kind of pull this stuff out? And what's the value returned? Maybe like, resources invested to value returned. And there's a lot of, I think, fair and criticism that this might be taking it pretty far out of balance.
Hey, here you go, Nels.
Yeah.
You make a wildlife park with all de-extincted animals. How's that?
Yeah, I've heard this plot line before. Jurassic Park. I think there's a new version coming out this summer.
What could go wrong?
Yeah, that's right. There's no cautionary lessons in any of this sort of history here, right? Exactly. Okay, so that's sort of the backstory, or that gets us to but how did they get to the starting line? That's where this preprint sort of, and what we'll spend a few minutes on. How did they get to the starting line? How did they know what changes to make? And we aren't going to quite connect this all the way to the final 20 changes that were made in those 14 genes, but this is sort of what got them to the starting line. So in order to sort of prioritize those 20 changes, and we don't know, like, were there 50 changes at one point, 80 changes, 100 changes? How did they whittle that down? But you have to know how is the extinct dire wolf related to modern canids? Where did it sort of emerge? What are the closest living relatives, or that might be like serve as sort of the framework, the genome, where you can't make all the changes, but you sort of prioritize ones? How do they kind of get there? And so The punchline of the preprint is that they sequence, assemble, and annotate two direwolf genomes using paleogenetics here. And these are from well-preserved tissue from two wolves that lived. They estimate that one of the samples is from about 13,000 years ago. The other is from about 72,000 years ago. So pretty long span with, obviously genetic changes happening as well between them. But this allows them to also determine some of the ancestry and how, like, sort of what actually are dire wolves most related to. And that revised sort of the previous ideas that were based on just really limited sequence. So this really expanded the amount of genetic material that allows you to then sort of weigh how these are related to species. And so in the
Nels,
end, yep.
why do they call them dire wolves? Do you know?
Yeah, well, I mean, that's sort of, I don't, but this sort of, that sort of also feeds into the, like, why were the dire wolves picked for Game of Thrones, right? It has this, and why is this sort of capture the human imagination? There's something about the, the branding here or the name of it. It really,
Yeah,
like,
sure.
right.
Okay.
Right. And so anyway, these big muscular white wolves with massive muscles, like in the dire wolf, just, I think that feeds into our, maybe evolutionary history with, as humans, we're coming up and competing, or predator prey interactions with wolves as well. It has that sort of underlying energy to it, or that thread to it. So previously, people or geneticists had put forward that maybe the dire wolf is actually most closely related to things like African jackals, other canine species. But this pushed, so 2/3 of the genome, they through a lot of analysis, they make the case that actually a lot of the 2/3 of the genome are contributions mainly from gray wolf, coyote, and the dehole, or the red wolf, which is found in Southeast Asia. So, I mean, canine evolution, dog evolution is absolutely fascinating. We've spent a little bit of time on TWiEVO thinking about it in the past. So, episode 85, teaching old dogs new genetic tricks. People have also thought about, so all of the anthropomorphic selection that humans have done in the domestication of dog species, selecting for all these behaviors that we now, is also this really tight connection between humans and dogs evolutionarily. And then using the dog breeds and all of the pretty, sophisticated genetics that have gone into that to try to understand the genetic basis of behavior, of mammalian behavior, that could even apply to us. We had another one not too far after that, Vincent, episode 88 of TWiEVO, Dog Days of Evolution in the Shadows of Chernobyl. So these are these kind of now semi-wild dog packs that run around Chernobyl. A study that looked at the long-term impact of exposure to ionizing radiation from the nuclear disaster there. We also, one of my favorite episodes that we did is not on the dogs themselves, but on the tumors of dogs, these transmissible tumors. This is episode 46 of TWiEVO. Can an old tumor teach us new tricks? Or you can see we're going back to the well here.
Transmissible facial tumors, right?
Well, that's the Tasmanian devil. These are
These
venereal.
are dogs.
Venereal tumors
Yeah.
that are passed by sexual contact. But here, what's wild about this? So using genetics and genomic analysis of these tumors, they trace it back to a single dog from something like 6,000 years ago, and then sample modern tumors, dog tumors from around the world, and start to trace the sort of an echo of dog evolution is in the sort of genetic differences in these tumors. One of the wild mutation patterns they see is that the matches the latitude. So that the more UV radiation that the dogs are exposed to, the ones that are living near the equator, the more mutations of that sort of UV induced mutations that they see in these tumors from the dogs that are living in those regions. And so pretty cool, like, genetic stories here. So again, I think this, the dire wolf also sort of taps into all of these human connections as well, and the interest. And in fact, Eleanor Carlson is one of the authors here who does a lot of work on dog sort of diversity as it relates to genetics. So anyway, so that was, that's sort of the first, I guess, kind of evolutionary step forward, is to get a better sense of what direwolves were and what they're related to. And so a little bit more background. So dire wolves were thought to be the most prominent canid species in North America up until about 13,000 years ago, when they are estimated to have gone extinct. And one of the reasons there are good samples is that there have been preserved animals in the La Brea tar pits, where people have sort of, they've gotten to the tar, got stuck in the tar, died, and then because of that, sort of preserved and allowed these scientists to get preserved DNA samples. And so this also, the advances here kind of rely on other work in the field, improvements in so-called paleogenomics. And so the two samples they're using here, one is from a tooth, and we see a lot of genomes now, like from ancient humans, for example. So like thinking about some of the work coming from Svante Pääbo's lab. In Germany, there's another big group in Copenhagen. There's some authors here from Copenhagen. I don't know all the connections, but S.K. Villeslev's sort of launched this massive group at the University of Copenhagen in Denmark that does a lot of the sort of technology development on getting DNA samples. And as we've talked about before, the real challenge here is sequencing, or using damaged DNA and getting sequence that's not contaminated by modern DNA. So for the case of the dire wolf, you'd worry about other making sure that there aren't sort of dog DNA samples in the neighborhood, or exposure to dogs even. And for humans, if you're looking at like Neanderthal DNA, the first entire Neanderthal mitochondrial genome actually turned out to be Svante Pääbo's genome, published in a high-profile journal, because this isn't fraud. These were early attempts, but they were sort of up against all of this contamination. And the field has advanced really well. It's still there's still these challenges exist, but sort of the pitfalls and the progress along the way has made this better. So anyway, the authors here get two samples, an incisor tooth from the Cincinnati Museum Center. Oh, I wanted to say, Vincent, or I'll get here in a moment. But so again, these collaborations, not just international sort of groups, but also, this fabric of science. So working with museums, working with other, not just university, sure. Right? This is kind of how science works, is having all, and the reason I'm kind of, maybe emphasizing this or pointing it out, is that this fabric of science is currently under attack. It's being ripped to shreds in various ways. All of this sort of careful work over decades to build these connections really feels like it's vulnerable, or in a lot of cases, under attack, or the sort of resources or the sort of incentives that are in place to do this kind of work is it's becoming harder and harder, even in just kind of almost by the week. It feels like right now it's really wild times. So anyway, able to get one of the samples, a tooth sample, where they were able to get really good DNA. This is the younger sample. It's about 13,000 years old, from the Cincinnati Museum Center. And then the other is a skull that they got were able to sample from the Idaho Museum of Natural History. So again, all points on the map here, and different sort of organizations that make this possible. In addition to the samples, then the next big step is, once you have these samples, you get DNA. You try to keep it as clean as possible without the contamination. If you get good samples, now you can start really sequence this to like the technology and sequencing is really advanced as well. So they get almost 3 billion reads from the tooth. That ends up with about 3.5x coverage genome here. Yeah, exactly. From the skull, small piece of the skull, they got more than 33 billion DNA reads. That gives them almost 13x coverage for that genome. And so here, not just advancing that technology, but sort of practicing that sequencing often depends on core facilities, DNA sequencing facilities, which is what was the case for these samples, again, under attack. So when we think about, these are kind of also in the news headlines recently, was this notion that NIH grants are going to have what's called an indirect cost, right? So this is money that's set aside. So some of the money goes directly to the project that's being funded by taxpayer money. And then some of that money has been carved out in sort of indirect funds, which goes to the facilities and things like core facilities that are funded by these sort of like sequencing centers. So everyone benefits in a department or a university if there are these resources available to, if you do all the hard work sequencing, getting the samples, getting the DNA and then actually getting the sequence. These are, again, it's a handoff. This fabric of science moving forward. If you undermine and cut off the indirects or drain those down to 15%, a lot of this work goes away because those core centers close down. They don't have they depend on these resources, these indirect funds, to keep running. And so that's kind of another facet of the story I really wanted to spend a moment on, is to point out that although it's sort of colossal biosciences, there's sort of at the forefront of promoting the work, and they have a pretty big, PR staff that's teaming up with things like reporters from Science Magazine or Time Magazine, et cetera, other sort of press outlets. It's the deeper you go, it really depends, or they're taking advantage of, in good ways, all of those resources, all of that science that's out there. But that kind of gets pushed under that you don't see that part of the story, or maybe recognize the kind of side effects or the impacts of, as we are in pretty jarring ways reorganizing how we support or fund science. This all goes away.
That's why this whole blanket cut indirects to 15% makes zero sense, because they don't understand why it's very useful. It's just a punitive measure, and it's just, to punish things that are great is ridiculous.
Yeah. Well, and so that's why it's worth it to kind of, as we're telling these stories of science on TWiEVO, to try to bring that forward, because we don't often talk about that. And I think, in some ways, myself included, scientists sort of take it for granted. And so we can't take things for granted anymore. And so I think we need to really illustrate sort of how the sausage is made in science, and how many connections and sort of interactions that depends on, and how important and vital our taxpayer resources are in this pursuit. And so whether or not we think this is a good use of resources, as we kind of unpack where the technology is being advanced versus sort of the provocative headlines, underlying all of this is this incredible system in the US that has been built over decades and is now in real jeopardy.
Well, all of these cuts, from grants to indirects to everything else, are being made with no appreciation for the end result. None whatsoever, because no one has done any research into seeing what should be cut. They just cut, right? And for example, the FDA used to assure milk quality, right?
Right.
But now they fired all the people who do that, so they can't do it anymore. How stupid is that? If you think something is essential, you research it and you figure it out, right? And they didn't bother.
Yeah, I agree. No, this is a lot of fire, ready, aim instead of ready, aim, fire. There's also, of course, the first thing that's sort of put out front is this notion of inefficiency or something like that. And is it perfect efficiency?
It's just a lie.
It's a lie. I also wouldn't say that science is perfectly efficient, or that government is perfectly efficient as well. But I think where the big disconnect here is that it's this idea of, well, here, identifying this one thing that seems inefficient and then pulling it off the rug as if everything else is okay. And, maybe an analogy, and it's just not, because this is all woven together. So if you have a handcrafted woven rug, and you say, this gold thread in the middle here, I think that's inefficient. It's not so important. So let's pull that out. The problem is the entire rug that unravels and comes apart. And so we still want a rug, but if you, the way you do this matters. Exactly what you're saying, Vincent. And it's so chaotic and ham-handed that
taking away grants that have diversity in them is not for efficiency. It's just ridiculous.
No. Right? Oh yeah. Well, that's a whole nother topic, a related topic at play here, which is also really destructive.
For years, it's been required to talk about diversity in a grant application. Now, all of a sudden, the rug is pulled out from under you, because this is completely unfair, totally unfair. And we have to sue, because this cannot stand. None of this can stand. In fact, it's all just petulance. When you think about, the universities getting punished under the guise of preventing anti-Semitism, and that's fine. I don't think there should be any such thing. But then to want to control over admissions and faculty, what does that have to do with anti-Semitism? Right?
Yeah. Oh no. Yeah. No, the means or ends justify the means kind of disconnect here is really glaring as well. I agree. On the fairness side, we are seeing some lawsuits being advanced. So that 15% indirect, which is illegal, it's going back on a law. Commitments has been largely blocked. Of course, now the new budget is kind of coming forward. We'll see how that gets renegotiated, et cetera. But yeah, absolutely right. Good to see the loss. Good to see since our last episode, obviously the biggest sort of news headline is Harvard standing up, but with a lot of
Yes.
partners and other universities sort of pointing out how everything happening here is totally illegal, and not just sort of giving in or negotiating, but actually sort of standing up for science as it's practiced, has been
Thank
practiced for
goodness. Thank
decades
goodness
in the U.S.
Harvard
Yeah,
did that,
yeah,
because
agreed.
otherwise
Yeah,
everybody else would be caving, and that's not the thing to do.
yeah, I agree. And now
You
I
shouldn't
think we're
give any
seeing,
you shouldn't give
yeah,
any of this up, because, as you said,
no,
it's all unconstitutional to be doing this. To say you
yeah,
want no anti-Semitism and then to say you want control over admission, that's ridiculous.
that's right.
When
Yep.
you're
And
exerting
so,
your power, you say, I'm the president, I can do anything. And that's just not true. You can't do
yeah,
everything,
no,
right?
that's right. Agreed. So there's, and not just Harvard. Good to see now. I think I saw just in the last day or two, 150 presidents of universities signing a letter, sort of in that same space. I might circle back. I'll probably have two or three picks of the week at the end here. One of them is a great interview with the president of Princeton University. This was
Hmm.
on the Daily Podcast about exactly this. How do we organize and stand up? Sort of contrast that to what happened at Columbia University. No offense,
Yeah.
Vincent, to your alma mater, so to speak, but yeah, and it's complicated, and different universities at different times might make different calculations, but as a whole, this notion of we got to stand up for science, and good to see at least some efforts in that direction.
It's not my
Okay,
alma
so
mater. I work there, Nels.
I know, so to speak. It's important.
I was
Yeah,
always
important
upset about
distinction.
their position, right? They
Yeah,
were
of
just
course. Yeah,
caving. And,
exactly. Yeah,
if you cave, then they'll come back with more stuff that they want. So
yeah, that's right.
I'm
Yeah,
thank goodness they now have an acting president who's going to stand up to a certain extent. But it's because of the president of Harvard. Yeah.
yeah, and others out there. I think, yeah, but certainly that one's one of the most high profile examples, and that matters. That it really does. Okay, so back to our story. So we did kind of a stem winder there on the reason they could get all these great sequences is because of this fabric of science, or these core facilities that
Yeah.
they leaned on in order to get this. Okay, so then a lot of work next to not just get the sequence, but then to assemble it, to make sense of it, especially as it relates to these samples you have all of this issue with the DNA degrading, the quality of the samples, weighing that against contamination. So one of the innovations here is they use some new algorithms, these iterative techniques. So first of all, now that they've kind of gotten a sense of a lot of the sequences related to gray wolves, they use that as a sort of framework or a template to compare the genetic changes on. And then they keep sort of iterating this algorithm to try to get better and better assemblies that sort of take into account both the relationship between the other species, but also sort of the quality. And it's kind of it's not going to be able to do it justice here. And they do note that this isn't perfect. So if you sort of iterate and you kind of make some mistakes early, that by continuing to build off of those early mistakes, you might sort of make that mistake even worse in terms of, you think this region of the genome or this part of the chromosome has this sequence, but if that started kind of off on the wrong foot, it can really go off the rails. So you need to take some of this with a grain of salt, but that is, a real advance here. And again, as we're kind of going beyond the headlines and into what is the value here, paleogenomics is improving. All of the energy and resources they're putting in here relates to that. So with the gray wolf genome as a template, they note almost 15 million genetic changes. So 13.5 million of those changes are DNA mutations, single nucleotide variants. This is, the A's, T's, C's, and G's are a different letter at a given position. There's also nearly 400,000 insertions. So this is new genetic sequence that's been added, and that could be as little as, a couple of base pairs of DNA, a couple letters, up to massive regions that are inserted into the genome. And then more than 500,000 deletions, where sequence is lost as you compare the gray wolf to the dire wolf. So this is sort of an important moment for kind of, again, defining de-extinction here. This is not a dire wolf genome. If it was, they would have to make 15 million changes or sort of synthesize this thing from scratch. They can't synthesize from scratch. They don't have, I think in the end, they have about 90% of the genome. So they really need a starting template. They use the gray wolf, which is, again, they made, I think, the right choice. That's probably better than an African jackal, given the analysis here. But then, there's a big sort of difference between 15 million changes and 20 and 14 genes. And so this is not a dire wolf. It's not even close, just right in terms of,
Yes.
yeah. And so, but they prioritize on some traits that they think sort of, underline sort of the interesting differences between the dire wolf. So we'll get to that in a moment. But first, they base, on the mitochondrial genomes that they put together. They can then propose a phylogenetic tree with existing canids or dogs, wolves, etc. And so that's in figure 3. So it doesn't, and even here, canid evolution is complicated, and so it doesn't resolve these relationships. There's a really complex history of different breeding, introgression. These are genetic contributions, sort of from different sort of populations and ancestries of dogs. One of the things they can do, though, they have enough sort of signal in the data to be able to say that instead of the last common ancestor as dire wolves, as the species split off from other canids. So earlier work suggested that might have happened 5 or 6 million years ago. The data they have here suggests it's actually more recent, 3 million years ago. And that it's sort of a closer relationship to those gray wolves. And so that, again, sort of gives you that, getting you to the starting line. It just makes it more feasible to use the gray wolf if it's 15 million changes, but it might be 30 million changes if it really were 6 million years ago. And so to be able, maybe it just raises the possibility of making some of those changes actually having sort of an impact. It's not sort of ancient evolution, it's sort of more recent evolution. And so using that template, has the sort of potential of giving some of the traits that you're hoping to see. And then let's talk about the traits. So again, a little bit of a disconnect, to say the least, between the preprint or the information they've given us so far, and then the 20 changes that they make in those 14 genes. I don't know what those 20 changes are. I don't know what those 14 genes are. But one of the things that evolutionary geneticists are always looking for is like signals that these changes of those 15 million, how do you prioritize? What are the important, sort of handful where it has a natural selection acted on this? It might have an impact on that species as it adapted before it became extinct. And so they look for these patterns. There's ways of doing comparisons. So they take their genome, compare it to 10 other genomes, and then ask, what are the regions that are changing in ways, genetic changes in ways that look like it's adaptive? They find 80 genes that they say are under positive selection. We've talked about this many times on TWiEVO. This idea that a genetic change isn't just, or a mutation isn't deleterious, doesn't make you worse, like we think about most mutations. Probably the vast majority of mutations are sort of relatively neutral. They don't kind of impact fitness one way or the other, but there are these semi-rare mutations where that change actually is beneficial. This is adaptive, the possibility of adaptive evolution. And so they find those kind of signals in about 80 genes. And these might be some of those changes that were engineered into the genomes of the wolves that became Romulus, Remus, and Khaleesi that are running around on this sort of private wolf resort, 2,000 acres at some undisclosed location in the U.S.
I mean, I think in the preprint they talk about some genes involved in body size, and I would guess that's one of them that they could put in because you could measure that right away, right?
Yeah, exactly. So they mentioned that gene on body size. In the end, I think gray wolves are kind of big too. So it's, I think there's maybe a little bit there. And then also, again, sort of this tapestry of science, right? So they, so what are the genes that go into coat color or hair color? We've talked about this, I think most recently about cats, calico cats. And so they can use, they have this sort of encyclopedia of developmental biology where they can kind of cross list, okay, here are the genes that we think are important for things like coat color. Here are the changes in those genes that we see in this genome. Some of these cases, it might be these ones under positive selection. Other cases, they might not. I think a lot of the ones they see here are involved in fertility and reproduction.
Yeah.
And so if or how that came into the story, I'm guessing it probably didn't, but that's not too surprising. So usually the categories of genes or the kinds of genes that are adaptive are the ones that sort of like, how do the genomes get into the next generation, right? How do these populations compete? How does this genetic material move forward? And so the two big categories here for adaptive evolution are oftentimes reproduction and also immunity. How do you survive the infections that might otherwise mean that you don't have offspring? Those are kind of the two big categories. They don't mention immunity so much, but they do mention reproduction, and so that's probably a key sort of part of the story of the
Yep.
rise and the
Yep. Yep.
fall of the dire wolf. Um, okay, so back to so that's it. So that's where they leave us, right? Is with two, like, a lot of work, taking those two bone samples, skull sample, tooth sample, sequencing, assembling the genomes, and doing these evolutionary comparisons, and then getting a gene list that then they did a lot more work with, to filter down to those 20 changes. All of that, we're still, as sort of the science hungry public, we're still in the dark about what are the sort of techniques they used, other than the information that you can get from sort of cobbling together Time magazine articles, et cetera. Not awesome. So I get it that, oftentimes companies have intellectual property that they want to protect. But I think, for my money, if you go this big in terms of touting, sort of trying to grab everyone's eyeballs and attention, it would be nice to have a little more of these details on the record. And maybe that's forthcoming. I don't know. But obviously this is now well behind. We're now a couple weeks on, without really knowing what's under the hood here, in terms of a lot of the important details. Okay, so let's get back, Vincent, to the controversy. So that's kind of it. But let's talk about, I want to close, I guess, on a little bit more of the controversy of this idea of de-extinction. So Time magazine calls this the return of the dire wolf. Highly coordinated reveal here. But, I think some really valid criticism coming from a lot of coroners, a lot of scientists who care about this deeply, and want this technology to be developed and used in positive ways, but sort of the disconnect between that and then sort of the way that this story has been reported or sensationalized. So to put this in a little bit of context, it's been 30 years since Dolly the sheep was cloned, I guess back in the mid 90s. It doesn't, I can remember like another sort of splashy advance in terms of, right. And it's important to note that the technology here is still pretty crude. A lot of these animals that are cloned, and there's been a lot now. I think there's more than 1,500 dogs that have been cloned. There's a lot of pet owners that become really attached to their specific pet, and instead of breeding the next generation, or finding a dog of the same breed, just can't let go and want that same dog. And so rich people have been cloning dogs, so it's the same dog, right? Or it's not the same dog, but it's the same genome, or they start with the same genome and then it goes on. But doesn't come without problems, health issues, reproductive issues, et cetera. It can be really, I think, probably even more heartbreaking as you try to hold on to something versus sort of letting genetics just do its natural thing through the generations. But this is also probably most important, sort of in terms of human sort of efforts in the areas of livestock in agriculture. So a lot of pigs, horses, goats have been cloned since Dolly, other sheep, of course, but also mice, deer, cats, et cetera. And so this is, again, sort of putting this back into that context. That's where taking the blood samples, these endothelial precursor cells, sort of makes those possibilities or offers new pathways that could be really useful for doing better in that space. Some additional criticism, though. So there's a great article just this last week in Science Insider, the author Christine Wilcox, who is kind of trying to take all of the feedback and sort of put that together. So she got a quote from some paleoecologists, so ecologists, like thinking about the ecology, the ecosystems of the past and how that relates to modern ones. So this is Nick Rollins and then the ecologist Michael Knapp. They wrote a piece about, and one of the quotes here that I thought was really important, saying that they'd argue, this notion of de-extinction for this idea to be true, or even the concept. So, let go of the 15 million missing genetic changes for a minute, but just sort of give them the idea that these sort of look alike or behave or could potentially behave like dire wolves. They note that those pups need to function like dire wolves in an ecosystem that no longer exists. Who's going to teach these pups to behave like a dire wolf? And so this is right. And so that's not happening currently. These wolves are living this sort of wolf resort lifestyle. They're not hunting or anything like that. They're being fed this sort of, really kind of nutrient rich diet. And credit to Colossal, they're taking care of the wolves. We're putting these resources into making sure that they're healthy, or trying to. But there's all of these, the deeper you go or the more you think about it, there are all of these disconnects. And so, you know, why did the dire wolf go extinct in the first place? I think the best ideas out there is that a lot of the prey, the megafauna, the large prey species, went extinct before the dire wolf. And then the gray wolf kind of moved into a lot of these niches, and were able to outcompete the dire wolves. And so if you were really kind of thinking about the, paleo ecosystem or the paleo ecology, you would also want to put back a lot of the megafauna. And so that as you kind of go deeper and deeper, scratch the surface, it becomes more complicated in a hurry. And I think, the notion that humans are just going to do a couple of cute tricks genetically and sort of will go back in time to the ecosystems of the past is kind of a farcical idea. There's also, I mentioned this, this convergence with the politics. So Beth Shapiro, who's the science officer at chief science officer at Colossal, had a quote that Christy Wilcox put forward. I think Secretary Burgum, this is the Secretary of Interior's quote, has been unfortunately politicized, and the spirit of his message has been lost. In our meetings with the Secretary, my takeaway has been that he sees immense value in the use of innovation and technology in the recovery of endangered species. A lot to unpack here. So first of all, some clear coordination between Colossal and the new Secretary of Interior. You can find these quotes from Burgum on the Colossal's website. And I think this starts to peel back a little bit where Colossal's coming from. So they're really trying to advance this technology, cloning technology, sort of in the kind of Dolly 2.0 space. But again, sort of this sort of potential clash then if we overhype the innovation and technology. Where are we in terms of recovering endangered species? I mean, I think the best case scenario, so that, an example of that is that white rhino, which is right on the edge. Do we have good enough cloning technology to take the cells or the blood of the last 2 or 3 individuals in this entire species, to reprogram the nuclei and the answers might be, we're not there yet. We don't have the technology in place to pull this off. It's, really low percentages, depends a lot on what are the donors, are who are the surrogate mothers. How is all of that sort of reproductive biology going to line up here? And so unfortunately, as we're sort of arguing about dire wolves, there's something like 30% of species in the world that are, in some estimates, might be gone by 2050 in the next 25 years. And so, this is a case where the technology might really fall short. And especially if you sort of just depend 100% on that versus the conservation efforts, the sort of old fashioned, less flashy, less showy, but, to me, really, are we doing an all of the above approach, or is this sort of politics where all of the above means, in the sort of energy industry means more fossil fuels? This feels like this feels like fossil fuel 2.0, or maybe nuclear energy would be the analogy, where there's
good idea
to solve a problem that you can't solve unless you're really sort of hitting it in many ways. So I guess the last quote, they're kind of connecting this back to the endangered species, right? So conservationists find this kind of idea from the Secretary of Interior not so reassuring, because the Trump administration is currently trying to redefine part of the Endangered Species Act. Destroying a protected habitat is no longer counting as harm under the proposed rule changes. Hopefully, the bureaucracy, the slow wheels of bureaucracy won't allow them to do, or we're all kind of bracing for how much damage is going to be done in the years ahead, and what that means, not just for science, but for other, important key parts of the fabric of society, the U.S. society, going forward here. Anyway, so I've got a couple of links that might show up in our show notes at some point, a good summary of the issues. There is a good article from the Bulletin of Atomic Scientists, actually, a biotechnology company claims to have resurrected the dire wolf, but then sort of weigh some of the issues that we've been describing.
Let me go through a few comments that we've had here in the chat.
Good idea.
Before we do some picks, Nels.
Sounds good.
So YWSL joins us from Jerusalem. Welcome. Good to have you.
Welcome. Yeah.
Tom says, thanks, Nels. This could be catastrophic for conservation biology with the
Yeah.
delusion that we can de-extinct species while we lose extant species, destroy habitat pell-mell.
Yeah, absolutely. If it goes, you can imagine sort of all extremes here, right? The best
Yep.
case scenario might
Yep.
be somewhere in the middle where we keep advancing technology, but we keep doing conservation biology. Yeah. Thanks,
Les
Tom.
Fabi says a breeding pair of animals does not bring back a species because
Mm.
practically a species is a population of animals, not just a pair.
Important, really important point here.
Yeah,
And
good point.
and again, the deeper you dig, the more things kind of from the surface start to fall apart a little bit or become less shiny the farther you go. Yep. Thanks, Les.
John asks if we
Hmm.
decided we didn't want this research to continue, could we realistically stop it worldwide?
No, and I don't think we should. So I think, the same way that we are exploring, advancing nuclear energy today, not great because you create this new problem with like nuclear waste, but the better, the more humans advance it, the more problems you solve, the more kind of new problems you create. So for me, this falls more closely when we kind of take away the dire wolf and the Game of Thrones kind of energy uh, in and around cloning Dolly the sheep and putting it back into that sort of more agricultural context. There's some real reasons where biotechnology or genetic technology and putting it into an evolutionary context can really, I think, be useful and help advance things. So I don't think we're going to stop it worldwide. I don't think we should. But what I think we should do is try to do a better job of understanding sort of the PR
Mm.
and especially as it relates to de-extinction, as that gets garbled up in conservation efforts, as Tom and others are pointing out here, to try to do better, to sort of find sort of the right places to put our attention and energy, or how to react in other sort of facets of society that don't make things worse instead of better. Yeah.
Let's see. Another one. All right, here. Rob says prospect of bringing back more Neanderthal with
Mm.
politicians is terrifying. That's true.
Oh my, yeah. Well, just bring back Neanderthal. So this has been floated as well. Nick Wade, who is a controversial former New York Times reporter, um, I can remember a quote. I know this one's stuck in my craw from a few years back, a decade ago or something, saying that we should use this technology to make Neanderthal armies to, like, basically do the work for humans while other humans sort of sit back and with their feet up drinking cocktails or something. The ethics here go off the rails really quickly, especially as we bring this into sort of human biology. And then ideas of race and racism aren't too far behind in a
Mm.
lot of these conversations. And so, yeah, terrifying and more than that. But again, so I think not terrifying in the sense like let's bury it and not talk about it, but in the sense of, okay, why do we really need to understand genetics, evolution, the implications for that, things like, we had a great interview. The author, one of the co-authors of the book, "Racism, Not Race," and this is
Right.
in this moment in particular feels really important to go beyond the news headlines, to go really deeper into what does the biology mean, and to try to share that with each other, as we're all learning as we go, to understand the implications here, and then and see how that impacts. That has impacts beyond just the genetic science.
John writes, I'm frequently astonished by things that can be done. How hard is nuclear transfer? Seems crowded in those cells. So how do you even do that?
Yeah, and those, like, kind of fundamental advances are now 3 decades old, and so and that's one of the ongoing challenges here, right? Is, so first of all, what are the cells that you take this from? The more flexible you can be in that, that's a real advance. And then do you bring up the frequency of success? And with the side twist of doing genetic engineering along the way. So another example of this in pigs is, growing or genetically modifying pigs, excuse me, potentially making clones for things like heart transplants. And so George Church is sort of is involved in this, along with other many groups who are trying to do this. One of the early sort of failures along those lines was there are all these endogenous retroviruses in pig genomes, and so they ended up making 60 edits to try to get those offline,
Right,
so that you don't,
right,
when
right.
you don't get right, because if you get a transplant, but then you just sort of activate all of these viruses in a sort of unfamiliar immune system, that could have all kinds of side effects and implications as well. So yeah, logistically, biologically, genetically, it's very crowded in many different ways, and so all kinds of technical hurdles here. And so yeah, this is the work is good and is being done in many facets. Um, uh, no question about that. The ethics catch up with you very quickly, which for me really underlines why. So I'm in Utah, where the state legislature has been getting more adventurous in attacking things like the liberal arts. We should
Yep.
only be
Yep.
supporting, hard sciences or engineers or STEM in ways where the graduates have high salaries. We should be supporting the liberal arts, where we take the technology that we're developing, put that into the broader context of ethics, philosophy, humanities, and ask ourselves, how do we want to deploy this technology as it continues to develop? That's really, to me, the best prospect of things, or there are going to be mistakes and there's going to be disasters along the way. But to mitigate those and to put the technology to its sort of, closer to a more positive or beneficial outcome.
M.E.
Great points,
Wheeler
Chuck.
is checking in from Helsinki.
Fantastic.
Welcome.
Yeah, good to have you back. Glad you're here.
Cheyenne wants to know if the DNA of mitochondria of eukaryotes are related to prokaryotic DNA.
Yeah, good question. This is another favorite topic of TWiEVO, going all the way back to the origins of life. And so I don't have at my fingertips some of the episodes, but we've been weighing a lot of these really cool deep sea discoveries of archaeal species that help to sort of think about the relationship between um, prokaryotes or bacteria and archaea, and then eukaryotes that at some point in ancient times gained an entire endosymbiotic events, gained entire and ancestors of modern bacteria slash archaea, and then came online to then degrade or become today's mitochondria. So yes, there's some ancient connection. Not only is there a relationship, but this was like basically a predator prey relationship where an ancient cell was taken up, and then that became the modern mitochondria billions of years on, or billion plus years on. Yeah.
Patrick is from Puget Sound. Welcome, Patrick. Hope you enjoyed this. Andrew says, percentage-wise, it seems like changing only 20%, only 20 genes wouldn't matter if they use corgis as the base. Then, if they wanted to be overly dramatic, they could call them sure corgis. Dire corgis, that should be.
Right. Dire corgis. Well, great. I love this thought experiment, right? So yeah, if you make the equivalent of, you could do that, technically possible. What would these look like, dire wolves, or would they look like dire corgis? Probably dire corgis, right? So they might be white. They might have thicker coats. They might be a little bit more muscular. Although it's a great question, because or a great idea, raises the question of whether or not, that template genome that you start with and the relationship between the species, is there enough sort of additional distance, including in the anthropomorphic or the human-based selection of corgis, all of the selective breeding, that when you make those changes to the equivalent genes, first of all, are some of those changes exist? Do those genes exist? Are they deletions, insertions? That's all sort of in play. But if we just assume that you can make those 20, will they have the same phenotypic impact? Will it, coat color, et cetera? And so, yeah, but it's a great way of, I guess, a great thought experiment, because if these were dire corgis, like how would the public react versus the dire? Like, again, it's sort of feeding into the human imagination as part of the promotion of the work. That's a great one.
Thanks, Kent, for your contribution to science communication.
Yeah,
we
appreciate
it. We need it.
Yeah.
And one more comment from End Root. In D&D style games, they often use dire as an adjective for large muscular subspecies of animals. A lot of people joke about D&D farmers raising dire turkeys because of their superior size.
Yeah, I love it. That's great.
You ever played D&D, Nels?
Oh yeah, way back in the day, like kind of early teenage years, but haven't recently.
All right, Nels, let's do some picks.
Good idea. Let's see here. So, my pick of the week here is, so I'm going to have my supplemental pick. So I don't have the link at my fingertips, but there's a great interview with the president of Princeton talking about standing up to some of the attacks on universities, on science funding in particular, and the disconnects between the sort of ideas put forward of why the motivations for doing that and how sort of illegal or off base that is. It's on the Daily podcast, maybe from a couple of weeks ago or a week or two ago. But my sort of official science pick of the week is um, a short article. It's actually an advertisement for the Vilsec Foundation. So the title here is Navigating to the Forefront of Innovation, a Celebration of Immigrant Scientists. And so the Vilsec Foundation for years, I don't know how long, have been giving out prizes to scientists or scientists, engineers, physicists, et cetera, who've been practicing science in the U.S., but grew up or were born in different countries and then came here, immigrated here. And it really highlights, again, sort of using this analogy of the fabric of U.S. science, how international it is and how how immigration is at the core of the energy, the innovation of science moving forward. So at this link, 4 great examples. This year's prize winners, um, just doing breakthrough level work. I have a sort of indirect connection to this. So my former postdoc mentor, when I was kind of coming up before I opened my lab, received a Vilcek Prize when I was there. This is the early 2000s, Harmeet Mullick. And I'm just remembering, how much fun that was to have that sort of association with Harmeet, him receiving that prize. They also give prizes to like chefs and things like that. And so we all went out for a lab lunch, and it just had this, great kind of inspirational energy that in the US, people come here, train, open labs, do incredible things, sort of grow scientific careers. And over decades, this has just been an absolute engine of scientific advances. And so that too feels like it's under stress or worse as we're reading all of these horrifying stories in the newspaper about visa holders, immigrants losing, being, starting the process of deportation in some of these cases for pretty flimsy at best reasons. In some cases, it's becoming clear that these are just out and out mistakes. They're just sort of, they're making sort of whatever errors in their data sets or all kind of the Doogie or whatever it is related efficiency garbage. And it is just that, like, read this, the Vilcek Prize winners and what they've been doing scientifically, strengthening this country. Compare that to this sort of garbage time of just kind of semi-randomly kicking people out of the country who are trying to pursue scientific careers. And it is an absolute just contrast in, sort of human efforts. On the one hand, completely inspirational, engine of discovery innovation. On the other hand, just a bunch of lazy people being just sowing chaos and being mean. Really unbelievable. Anyway, so that's my science pick. I'm trying to stay positive to celebrate these prize winners, but you have to look at today. You have to also look at the other side of the coin, which are the attacks on people just like this.
This country is built on immigrants, and to think that it should be run by white men of Christian conservative bent is totally ridiculous. And we need to change this, folks, because it ain't going to work. My dad was an immigrant.
Yeah.
He had a good career as a surgeon, lots of scientists, politicians, economists, geologists, cooks, everything. They're immigrants. That's what makes this country unique compared to any other country in the world. It's got a huge mix, and these guys want to mess it up. How do we get to this point?
I know. It's absolutely wild. And as has been pointed out, science is one of the U.S.'s best exports as well. And so as people are less welcome or under, attack or under deportation orders, all of that goes away, or a lot of that goes away as well. I mean, it's just on every level. It's just absolutely galling. I think this kind of also gets to your science pick of the week, Vincent. What do you have?
So my pick is a article in Nature. Unfortunately, it's paywalled, but I can read a couple pages before the paywall kicks in. It's called "Exclusive: A Nature Analysis Signals the Beginnings of a U.S. Science Brain Drain." It's by Laura Udesky and Jack Leeming, and so they have this platform at Nature. It's called the Nature Careers Global Science Jobs Platform.
Yeah.
and they can go through it and look at what's happening. And they found that U.S. scientists submitted 32% more applications for jobs abroad between January and March than during the previous period in 2024.
Hmm.
And they have some nice graphs here, which show how the interest in U.S. interest in international jobs has gone up. And many countries, say they have a big interest. They're talking with president of Aix-Marseille University in France, and they're saying,
Yeah.
we're really sad about what's happening in the U.S. We want to try and help. We can't take everyone, but we'll take as many people as we can. I mean, you can get a lot of good scientists in different, but it's not the same. You've broken this conglomeration that
Exactly.
worked so well. So you can break it up and throw them in other places. People will have jobs, but it's not going to be the same.
Yeah.
Anyway, it's a
Well,
good article
yeah.
to give you that background.
No, that's great. And like those decades to build these systems, how quickly they can kind of dissolve. I mean, if I were, in government in a foreign country, I would be trying to put as many resources into science and recruiting scientists from around the world in this moment. There's sort of this strategic opening, but it's not. But an important point here, I think that you're getting to, Vincent, is that you don't just sort of hire 50 people, 100 people or whatever, and all of a sudden recreate something that took decades to build. So, but that's an investment, a longer term investment, where if we come back, 30 years, 50 years as sort of the next generation of science, it might be that it's Canada that's at the forefront. China is at the forefront, et cetera. There's some really important signs that that's already happening. Those longer term investments are being made while there's sort of this short term attacks on or very quick pace of letting go of a lot of what has been built over decades. Really wild how long that sort of contrast between how long it takes to build something versus how easy it is to tear it down or how fast you can tear it down. Yeah.
All right, that's TWiEVO 113. You can find the show notes at microbe.tv/TWiEVO. We'd love to have your support, microbe.tv/contribute. And should you want to contact us and ask us something offline, TWiEVO at microbe.tv. Nels Eldies at cellvolution.org. On Blue Sky, L. Earlybird. Thank you, Nels. Good to see you again.
You too, Vincent. Great to be together. Do you have an office hours coming up tonight? Are you back in the game here? Yeah.
8 p.m. office hours.
Nice.
And between now and then, I have to teach a virology class.
Awesome.
Yep. Stay busy. It's good.
Good call.
I'm Vincent Racaniello. You can find me at microbe.tv. The music you hear on TWiEVO, let's turn it on. Well, let me thank our people.
Yes.
First, I want to thank everyone for being here today. You ask good questions, always love it. I want to thank our moderators, Barb Mack, Les, Tom, and Vanity Nutrition. Thank you so much for coming and keeping it civilized. We'll be back in about a month, right, Nels?
Yeah, looking forward to it. It'll feel like a year, and it'll probably be a year's worth of news to filter through, but we'll be here.
So that music, let's turn it on. Trampled by Turtles. You can find their music at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast, the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.